This is the lecture for Lab 10, Cellular Division. The cell cycle is the phrase we use to describe all the different stages a cell goes through in its lifetime. And the cell cycle is divided into two basic stages. The first stage is called interphase, and interphase is where most cells spend most of their time. And this is where the cell is doing its normal function. So if it is a muscle cell, it's contracting. If it's a neuron, it's sending signals up and down the body. The second stage is the mitotic phase. And the mitotic phase is when the cell is going through cell division. Mitosis is a type of cell division that is referred to as asexual reproduction. And it's referred to as asexual reproduction because it generates two identical daughter cells from one parent cell. And these cells are identical because they have exactly the same number of chromosomes and express the same types of genes. All cells in the human body divide by mitosis, with the exception of the cells that produce the eggs and the sperm. They divide by a special type of cell division called meiosis. Mitosis has 4 stages. Before mitosis occurs, the cell will be in interphase, and interphase is where most cells spend 90% of the time. But during interphase, there are 3 different stages. The first is called G1, and G stands for gap. And during the G1 stage, this is where the cell will get the signal to divide. And the cell will begin to grow, to get larger in order to prepare for cell division. Once it has grown large enough, it will enter into S phase, and S stands for synthesis. And this is the stage where the DNA is copied. So all the chromosomes are going to be copied so that both daughter cells will have exactly the same set of chromosomes. The last phase in interphase is G2, gap 2. And this is where protein synthesis will occur to prepare for cell division. So going through the diagram, again, most cells spend most of their time in interphase. But during interphase, there is G1. G1 is gap 1, and during this time, the cell is going to receive the signal to divide. Once it receives that signal, then it will go into S phase, where DNA synthesis takes place. And so every chromosome will be duplicated, will be copied, and then you will have 2 identical chromosomes. Once that has been accomplished, then the cell enters into G2 phase, where the rest of the cell gets ready to duplicate. And once that happens, then the cell will actually enter into mitosis. One of the important goals of this lab is for you to be able to recognize cells that are in different stages. When a cell is in interphase, a eukaryotic cell, you will see, has an intact nuclear membrane, and the chromatin, the DNA that is inside the nucleus, is unwound, uncondensed, and so it will have a nice even color. Also, it may be possible to notice the centrioles, and the centrioles will be at one side of the cell. During interphase, the cell received the signal to divide. It duplicated all of its chromosomes, and then it grew larger, preparing for cell division. Once all of that has been accomplished, then the cell will enter into prophase. And during prophase, the chromatin is going to condense into chromosomes. The nuclear envelope will begin to break down. The nucleolus is going to disappear, and the mitotic spindle is going to form. So cells that are in prophase, and there are 2 types of prophase: early prophase, so this is an example of early prophase, and late prophase. So what is going to happen is the chromatin is going to condense. So you're going to start seeing these colored bodies. So instead of a nice even color in the nucleus, you'll start to see what looks like little strands or pieces of yarn. That is the DNA, the chromatin, condensing. Also, the nuclear envelope is going to break down, so you're going to go from an intact nuclear envelope to no visible nuclear envelope. The nucleolus is going to disappear, so the nucleolus is where rRNA is made, or ribosomal RNA is made, so that will disappear. And the last thing is the mitotic spindle is going to form. So the mitotic spindle comes from the centrosomes, and those are the cytoskeletal fibers that are forming that will eventually attach to the chromosomes and pull them apart. So all of these things are preparing to separate the two copies of the chromosomes. So in order to separate them, you need to condense the chromatin, break down the nuclear envelope so you can pull the two sets of chromosomes to opposite ends of the cell. And the mitotic spindle is forming. Again, the mitotic spindle is going to be how these chromosomes are separated from each other. After prophase, the cell will go through 3 more stages, and those are metaphase, anaphase, and telophase. And here are examples of what is happening in the cell at these 3 different stages. During metaphase, the chromosomes line up along a central axis. So they're lining up along a central axis, and you have the spindle fibers attached to the chromosomes. And metaphase is a very short stage. After metaphase, we progress to anaphase. And anaphase is where those 2 sets of chromosomes are pulled apart. So that line of chromosomes is going to be pulled apart in 2 directions. So sometimes it can look like you have 2 lines of chromosomes or 2 groups of chromosomes that are moving toward opposite ends of the cell. And also you can sometimes see spindle fibers between the 2 sets of chromosomes. Telophase is basically the opposite of prophase. During telophase, the nuclear envelope is going to reform, and the chromosomes are going to uncondense, so they're going to unwind. Also, the spindle fibers begin to disappear. And the last thing that happens is cytokinesis, and cytokinesis is when the rest of the cell divides. So that is where the rest of the cell divides, and at the end you end up with 2 daughter cells. In lab, you will be looking at onion root cells and identifying cells at the different stages of mitosis. And onion cells, of course, are plant cells, so all these cells have a rigid cell wall. And then they are eukaryotic cells, so they would have a nucleus, and here you can see a nucleolus. So this cell is in interphase. As you look through the onion root cells, you are going to try to identify the different stages. So here is a cell that is in prophase because you can see the chromosomes are condensing, they're becoming visible bodies. Then this cell looks to be in metaphase because those chromosomes are lining up along a central axis. And this looks to be anaphase because now you have 2 lines of chromosomes as they're being pulled apart. And you can kind of see the spindle fibers here. And here, this looks like telophase. Telophase, you have 2 separate areas of chromosomes, and they are uncondensing, so they are unwinding. And eventually you'll get cytokinesis with a cell plate forming between the 2 and eventually growing longer to separate the 2 daughter cells. You will also be observing slides of fish blastula. And of course, fish are animals, so animal cells do not have a cell wall. One of the things you'll have to do is identify a single cell. In this view, this is 1 cell, and this is a cell in interphase because it has an intact nucleus and the DNA is not condensed. It's a relatively even color. This cell is in prophase. So in prophase, the nuclear envelope is beginning to break down. The chromatin, the DNA, has started to condense, and you can see spindle fibers forming on each end of the cell. This cell is in metaphase. So that is in metaphase where the cells or the chromosomes are lined up along the center, and you can see the spindle fibers very well. This cell is just beginning anaphase, so you can see that line of chromosomes is just beginning to separate. And this cell is in the late stages of anaphase or the early stages of telophase. So where the chromosomes have been pulled apart to opposite ends and they're starting to uncondense, and the nuclear envelope will be forming. And this is a cell that has gone through cytokinesis. When animal cells go through cytokinesis, they pinch off and they pinch into two daughter cells. Also in this lab, we will start an exercise on biotechnology, and biotechnology is where we use microorganisms to produce products that we are interested in. One subset of biotechnology is called genetic engineering. And in genetic engineering, this is where we manipulate DNA, usually to get a protein product that we are interested in. The first biotechnique that we will go over in this class is PCR. PCR stands for polymerase chain reaction. And this is a technique that is used to copy DNA. For this technique, the first thing you do is make a mixture of the different PCR components. And these PCR components include a sample of the DNA you want to copy, primers, the nucleotides, which make up the DNA strands, a Taq polymerase, which is a DNA polymerase, which will actually synthesize the DNA, some buffer, and you mix all of that in a PCR tube. Once that mixture has been made, the PCR tube is placed into a machine, PCR machine, which will change temperature to facilitate copying the DNA. The actual process that occurs is that as the temperature increases to about 95 degrees Celsius, the DNA will denature. That means those hydrogen bonds that hold the two strands of DNA together will break. Then the temperature will cool so that the primers can bind to the two strands of DNA, and then it will warm up a little bit again so that the DNA polymerase can come in and make a complementary strand of DNA. And that is called one cycle. So cycling the temperature from a high temperature to a lower temperature up again to a warmer temperature. And for every cycle, the DNA is going to be doubled. The second biotechnique that we will talk about in class is electrophoresis. And electrophoresis is used to separate fragments of DNA by size. And for electrophoresis, you need two things. You need a gel, and then you need electricity. So the gel is a matrix that the DNA is going to move through, and you have little slots that we call wells, and the DNA is loaded into the wells. To get the DNA to move through the gel, we use electricity. So we run an electrical current through the gel, and that will cause the DNA to move down through the gel. And larger fragments of DNA will move more slowly, so it will take them a long time to move into the gel from the well. Smaller fragments will move more quickly, so they will be found further away from the well. This is a photograph of an actual gel from electrophoresis. And always when running electrophoresis, we use markers. These are samples of DNA with known sizes, known lengths. Base pair refers to those pairs of nucleotides. So these are the wells where the DNA was loaded, and the electricity ran this way so that the fragments of DNA ran down through the gel. And here with this marker, the smallest band is made up of DNA fragments that are 121 base pair long, and the largest band, or the band closest to the well, has the largest DNA fragments of almost 2,000 base pairs long. And these are other samples of DNA that were loaded into the gel, and these are the bands of the DNA fragments.